This is our Universe Revealed lecture series. We have lectures coming every other week on Tuesdays at 6:30. We've had 2 physics talks, so the James Webb Telescope, and then the last one was looking at photons, looking at the Hadron Collider in Switzerland and how big science, big data works. Tonight we're going to dive into biology, the love of my life. And I'm really pleased to introduce Ana Lidia Flores-Mireles. And just before we were talking, and it turns out her undergraduate was actually in marine biology, working on whales, but got so interested in bacteria and quorum sensing and how bacteria were interacting with each other that she then did her Ph.D. at Cornell University in microbiology. And then did a research scientist position at the University of Washington University in the School of Medicine in St. Louis, Missouri. And then is now the Hawks Assistant Professor at the University of Notre Dame. And her key research areas are thinking about microbiology, immunology, and infectious diseases. And so how do you get bacteria and that interaction with the host and moving from beneficial interactions to really negative interactions. And so I will turn it over to you.
Thank you. Yes, thank you so much. Well, thank you so much for this invitation. It's always an honor to present to the community because we do this work for you. Right? My work is really translational research, and we're trying to understand how to help society to mitigate infections. So the first thing, and probably you saw the title, is what is the most common hospital-acquired infections? But before even going there, for the people that are not really familiar with this, I just want to tell you that hospital-acquired infections are those infections that you acquire once you get to the hospital. And sometimes it's not really clear. Sometimes you can have the infection after you were discharged from the hospital, but these infections can be incubating there, and after 48 hours, if you get those infections, those are considered as a hospital-acquired infections. And again, another definition is any infection that was not the primary visit to the hospital, right? So that's the main thing. So now that we have established what is a hospital-acquired infection, what is the most common? This is the type of, right? So normally we have 4 different types of infections. We can have more, but these are the most important ones, right? The first one that we can see, it could be catheter-associated urinary tract infections. I'm gonna go to the middle so I can see both screens. The other one that we can have is central lines-associated infections, or also called CLABSI, surgical site infections, and ventilator-associated pneumonia. And if you were reading the news or were listening to the news with COVID, we have many people going with, they have to be intubated because they were really critical ill. And they were having basically these kind of infections, but guess what? The people that also have ventilators and cannot respond very well, they need to have a urinary catheter, right? So from these 4 infections that we have here, the most common is catheter-associated urinary tract infections. And it counts for more than 40% of all hospitalized infections in United States and Europe. But this can also change depending on the settings, right? You have nursing homes, so many people have urinary catheters. So why do you need a urinary catheter? So you can see this is the urinary tract, and the urinary tract is composed of kidneys. You have the ureters, you have the bladder, and the urethra. So the function of this system is to filter the blood. The kidneys will filter the blood and will produce urine. This urine basically is the waste byproduct, right? So then what it does is the bladder will hold the urine and will expulse the urine when you need it, right? So probably some of you want to go to the bathroom and you're holding it, right? So it's that way the bladder does, trying to keep things tight. But what happens if you get impaired? Well, sometimes you get impaired because you have neurological dysfunctions, physical trauma, urinary incontinence, you have surgeries, many surgeries, you will have a urinary catheter. I have a C-section, and I have to have a urinary catheter. So even things that you don't think about it, you will have a urinary catheter. And then we have another situations where you have to get some specific treatments that have to be through the urethra. So the function of the bladder may be affected by this. So it is when you need a urinary catheter. So urinary catheterization will allow to safely remove the urine from the bladder into the outside, into a bag, right? So, and this is the urinary catheter. And you're gonna see this image a lot because we do tons of imaging of actually human urinary catheters. You will see that this part right here actually is the one that goes to the bladder, and this balloon is inflated right here, so it will hold the catheter in position so it doesn't come out every time that you move, right? So, I told you that urinary catheterization is really a common procedure in the hospital. It's believed that 20 to 50% of the patients have one, and that, as I told you before, may increase in different hospital settings. In nursing homes, sometimes the whole population has urinary catheters, right? And it's a big market. Not only is it only a big market, but the urinary catheter is really important for you to help you to remove the urine, but also has a consequence. Just the placement of the catheter predisposes you to develop an infection. And we need to understand why. So when I started working in this, 10 years ago actually, the most common data that we get, it was clinical data. And it was which pathogens were infecting the bladder. But we didn't understand why. And now if you think that this is 40% of all hospital-acquired infections, don't you think that we need to understand what is happening? So from now on, I'm going to refer to these catheter-associated urinary tract infections as a CAUTI. And as I told you, just the placement of the catheter predisposes you to get an infection. Not only that, is it often leads to bloodstream infections. The pathogen will go to the blood, will go to different organs, and then is when it becomes really serious. And if the pathogen is multi-drug resistant, that means that the antibiotics are not going to do anything against the infection. It can result in mortality. The other thing that we have seen is the people that have the catheter for really prolonged periods of time develop bladder cancers. So it's a lot of issues. The main problem that we have right now in the clinics. We have many of these pathogens, the majority of them, except for one or two, they are classified as a major threat by the CDC. That means that they are multi-drug resistant pathogens, and even our last line of defense antibiotics, they are not doing anything against them. So this becomes a really serious problem. But the problem is not only that, is how we see these infections. If I ask you, what is a urinary tract infection? The majority of the people will believe it's a community infection that's mostly suffered by women. 50% of the women will have a UTI, but you don't need any catheter. You can have a UTI, right? Then 25% of these people will have recurring UTIs their whole life. That's the problem. We're treating these infections as all the other UTIs that we know. And I'm going to tell you why it's different. So the problem that we have is that we believe that all UTIs are the same, and they are not. Why? One of the things that we know is, as I told you right now, community-acquired UTIs or uncomplicated UTIs is mostly affects women. These ones affects men and women similarly. The other thing is that with uncomplicated UTIs, mostly young women or postmenopausal women is the one that are going to be affected by this. In catheter-associated UTIs, there is no age bias. If you have a baby with a urinary catheter or an adult with a urinary catheter, they are predisposed to develop the infection. It's just a matter of when. The other thing that we see is that the pathogens that cause the infection. In uncomplicated UTIs, it's mostly E. coli. And you always hear, I have an E. coli infection, I have an E. coli infection. In this infection, it's no E. coli. It's still the majority of the cases, but we have a fungal pathogen like Candida. We have another bacteria that is a different group from E. coli
Enterococcus, but you see, there is a diversity. So that means that these uncomplicated UTIs is not the same as a catheter-associated UTIs, and we're treating it the same. And that could be the problem why we don't have any solution to it. So when I start studying this and say, why we have this issue? Why nobody cares if it's the main cause of infection in the hospital? So the first thing that I thought, the catheter must cause something. It's not only a surface for the pathogen to attach, but it has to have something. Why we have all these pathogens that actually they are commensals, they are normally in your gut. Why from your gut can become a pathogen in the bladder? But they are no pathogens if the catheter is not present. So the catheter has to be present for them to be infectious. Something has to happen to the bladder. So the first thing that I did is to try to analyze the bladder. This is a mouse bladder, and it's really similar to ours. And I'm just going to explain really quick. Do you see all this folding right here? This is essential because your bladder will go from being so small and then to have so much urine, hold the urine, then you go back, you go to the bathroom, you empty, and it has to go back to normal. So it's really functional, it's dynamic. It's one of the most dynamic organs that we have. And you have a flush of urine, right, all the time. But what happens when we have a catheter? Right here, I'm going to just show you, this is the bladder epithelium. This part right here, we call it the lamina propria, this little pink part right here. And then we have the muscle, which is this. When we put a urinary catheter, the structure of the bladder changes. Now it becomes inflamed, and you're going to see it right here. Do you see this is the bladder urothelium? This is the really teeny tiny part right here. So that means that you have inflammation. So it's totally different. And we don't have these foldings anymore. So that means that your bladder is not flushing the pathogen out, it's dripping. You don't have the same force to remove something from there. So definitely the dynamic have changed. But this is one part. What happens to the pathogen? Let's look at the pathogen right now. For the people that are not really familiar with these graphs, basically what we're saying is that every dot was one animal that we infected. We catheterized and infected. This is 1 hour to 14 days. And if you see 1 times to the 6, that means that 1 million bacteria right there. 1 million cells in your bladder. If you just follow the trend, you see that the colonization increased with the inflammation, which is really weird because normally you have immune cells that will attack the pathogen, and we don't see that. If you look at the catheter right here, we see a really high community also in the catheter. And something that I told you before is that these infections normally will predispose you to develop a bloodstream infection. And we actually see that after this infection is really persistent, they can go to the kidneys, spleen, liver, and heart, which actually recapitulates what we see in the clinics. So if we put everything together, what we can get from this? This was 10 years. Actually, this year is my 10th year working with catheter-associated UTIs, and I think that we have moved the field forward so well because we're trying to understand what happens. The first thing that I show you is that these pathogens, normally, they go to the bladder, and they cannot colonize. And it could be because you have the flush of urine removing them. The urine doesn't have much nutrients. It's not like the bloodstream. And probably you have immune response. But if you put the catheter, now you cause damage. Why we cause the damage? Just imagine yourself. The bladder is really small. You put this, it's basically the catheter is this size, like this big. Your bladder is going around the catheter, and then you move. It's scraping the bladder epithelium, right? I always think that it's like a shoe. Put a new shoe, you're walking, and then gets a little bit tight, then you're scraping, you're getting blisters, you're getting bleeding. It's exactly the same thing that's happening with your bladder. Every time that you move, every time that you do something, this catheter will have the opportunity to remove some part of the tissue. And that's why you have this inflammation. So we have the inflammation, and something that we discovered that was key is that as part of this inflammation, you have this protein that's called fibrinogen. And fibrinogen is essential for you. It's important for coagulation. So if you don't have fibrinogen, you bleed to death. So fibrinogen will go, trying to actually heal the damaged tissue, and then when this is healed, it gets removed, right? But guess what? In the bladder, you constantly damage the bladder. So this signal of inflammation is always on. It's always a problem. We need to heal the problem. So fibrinogen gets accumulated into the catheter, and now the pathogen is what we discovered. The pathogen was not binding to the catheter by itself. It was binding to this protein. And now it will bind there. It will use it what we call biofilm formation. So basically, they will have these big communities. Not only that, it will grow. And also, they use these proteins as a nutrient. So you have a bed and breakfast right there. That's why these pathogens are being so successful in the bladder, in the catheterized bladder. But when you don't have the catheter, they are not able to infect. So now this is perfect, and they can disseminate to other organs, right? So this is just for you to have a visual idea of what I just told you. This is catheter from the mouse, and you can see the tubing right here. And the green part is the fibrinogen. We're imaging fibrinogen, right? And you can see by 72 hours, it's basically totally covered. What happens with human urinary catheters? What I told you was with mouse. But we need to make sure that our system actually recapitulates what happened in human disease. If you look at this is just an example. Remember that I told you about the balloon, and this is the tip. This is the balloon right here. This is the tip of each catheter. We have different materials, silicones, elastomer, latex. In all of them, you can see the green. That means the fibrinogen is being deposited on those catheters. It doesn't really matter the material. We have tried antibiotic-coated catheters from different patients. We have patients that have the catheter from 1 hour to patients that have the catheter for 49 days, and we see the correlation with time and concentration of fibrinogen. So basically, with time, we're giving these pathogens the opportunity to colonize. But now, how it looks the pathogen, right? I told you that the pathogen was attaching to this. This is an example. Again, this is the catheters from mice. You can see the green. In red, you see the pathogen. And you can see how they colocalize, how they attach to each other. So you have different pathogens right here, even the fungal pathogen. All of them are using similar strategy. So they are taking advantage of the inflammation that we have in the bladder. But how it looks in humans? We have several patients that have actually an infection. And when you get these catheters, you see this is the balloon again. This is the tip of the catheter. We look for fibrinogen, and we look for different pathogens, C. fecalis, E. coli, Candida albicans, or Staphylococcus aureus. All of them are infecting the patient in a similar way. They are binding to fibrinogen. So that means that everything that we have found in the animal model, we're able to recapitulate in humans. Why this is important? I told you that we have antimicrobials do not work, right? We're not able to eradicate the infection. We have catheters that they have antimicrobial coatings. They do not work. What about if we remove the proteins from the catheter? What about if we create a new catheter that prevents that protein deposition? If they don't have fibrinogen, they shouldn't be able to colonize. So that's actually what we did, and we did a collaboration with Dr. Caitlin Howell at the University of Maine. And we create this new catheter that is really soft, is liquid infused, and the idea was to remove the protein so that the pathogens cannot attach. We did mass spectrometry. I don't intend you to understand this, but basically, if you have negative numbers, that means that our catheter was able to remove all these proteins from when you compare it with the catheter that was not modified. So that means that we were able to remove basically all the proteins that can be attached to this catheter. So then the question is, okay, we're removing the proteins. Can we actually remove the infection? So these are the catheters that were not modified, and this is the catheters that are modified. You see fibrinogen, and you see the pathogen in red, and you see the colocalization. You can see that with these catheters, we were able to remove the proteins, and we were able to reduce infection. But not only that, actually our concept was real. You see where the pathogen is colocalizing? Only where the protein was. So suggesting that this interaction is essential for them. We did that with E. fecalis, that is one of the pathogens, with E. coli, with Klebsiella pneumonia, Pseudomonas aeruginosa, Acinetobacter baumannii, and our Candida, our fungal pathogen. So this material works not only with one, it works with all of them. And why this is important? Because if you give the wrong antibiotic, you may promote colonization by other bacteria. And we know that the antibiotics that we use doesn't do anything against fungal pathogens. This is the first strategy that has been developed that do not use antibiotics, and we were able to reduce infection. Not only that, remember I told you about the inflammation? With these new catheters, we reduce the inflammation too. So it's believed that the constant inflammation in the bladder is what causes cancer. If a patient has to have a catheter for really long periods of time in the ICU, in a nursing home, we may be able to give them a product that will help them to reduce the incidence of cancer. So with that, I just want to tell you why we do this. We care about our patients. We care, actually, we just saw with COVID that the patients that have to be catheterized, they were developing these infections even further, right? Like the hospitals that have more COVID patients, these infections increase. And we don't have the antibiotics right now to deal with these infections. By understanding the biology of how this catheter modifies the bladder, we were able to create something that is cheap. And if you think that is 40% of the cases of these infections in the United States and Europe, now think about other countries that doesn't have the same sanitation procedures. So this catheter is going to be cheap. It doesn't have an expiration date, and it can go anywhere. So we believe that this could be some of the future in preventing infections in the hospital. This cannot be only applied to catheters. It can be applied central lines. You have stents. You can have dialysis catheters. So anything that can probably induce this inflammation, get these proteins deposited, and allow these pathogens to attach. So with that, I just want to thank my lab because they are amazing and they work in different things. I just showed you just one part. We have worked with the immune response to see why the immune response is not helping, why the immune response is not actually dealing with infection. And we think that we finally figured it out. And if we can give the perfect treatment for the immune cells to be more active, and we have a new material that prevents this protein deposition, I think that this infection may be history in the future. With that, thank you so much.
Is it still being tested? It's not in use as yet?
It's not in usage yet, but we have the patent going. We have to get the patent first.
Okay.
And then with the patent, we have already people that we're contacting to get the materials, and then we can modify the materials. And we're talking with FDA and all these, there are a lot of regulatory procedures that we need to follow. But I have a really long-standing collaboration with the School of Medicine at WashU, and I work with a lot of urologists there. And they're really interested that as soon as we have the product, we'll have the first testing.
So is there a predicted in the future date that this might occur, or
hopefully
no,
soon?
Because
hopefully
it depends
soon.
on
Believe me,
yeah.
I'm the first one that want to move this forward.
Right.
But but we're trying to do our best to get it out as soon as possible.
Okay, thank
Yes.
you.
How do you think this was going to be accepted in the medical world?
So for first, the good thing is that I have a lot of interaction with urologists, right? So I know what is the main problem and what is the main concerns that they have. To the fact that we were funded by NIDDK, which is is part of NIH and gives funding for problems in the urological or in the urinary tract, right? They believe, and one of the main comments that we received, this is the best option that we have right now. So we think it's going to be well received, and mostly because our modification, at least what we use to modify these catheters, is not toxic. It has been used before in patients. So we believe there's not going to be a big issue. And I think it's going to be well received, mainly for the main issues that we have, right? The infections cannot be controlled. Yeah.
When I came in here, I would have said that the urinary tract infections acquired in a hospital were because of poor procedure. Was that taken into consideration?
So yes, and even in the, after 2008, that we have the Affordable Care Act, where they force the hospitals to be responsible of the infections that happen in the hospital, so the patient doesn't have to pay for that. Many hospitals have increased the safety and the way that they work, right, to trying to reduce the infections. But infections happens, right? And the problem is that we have the gastrointestinal tract so close to the urinary tract that even if you go to the bathroom, if there is a small contamination, that pathogen may go up, right? And it's just a matter of time. How fast can that go? And I didn't go into detail because it's just 30 minutes, but we have found that there is a specific patient population that are more in risk to develop this infection faster than others. And it happens to be that they have more fibrinogen deposition than other patients. So it could be contamination because even your own self, your skin has have microbes. The microbes can migrate up. So it's basically almost impossible to prevent these infections. You can remove the catheter, but it's not cost effective. And also the nurses are really burned out to be taking care of so many patients and removing the catheters daily, right? So, yeah. Any other questions? Mm-hmm.
Hi, Anna. My name is Ili Chen from IU South Bend.
Hi.
So I have a couple of questions that are
Sure.
related. First, I know that you have a patent
Yes.
going, so I'm just curious, what is so special of the surface of this catheter? And not only can discourage colonization by pathogens, but also the fibrinogen. So the second question is that I learned that you learned about, just heard that you have a marine biology background.
Yes.
So, people often make comparisons of a shark skin and a coral.
Yes.
Those animals live in ocean, but they don't have biofilm growing
Yes.
on them. So they often use the surface structure as an idea for designing antimicrobial material.
Yes.
So
Yeah.
that's my
So, so for the first one, why it makes this modification so special? So the first thing that we know, or that we have seen, is that it's really it repels water, repels molecules, right? So just by doing that, kind of hydrophobic, but not completely hydrophobic, it's really slippery. It's extremely slippery. And to the point that you don't even have to use a lubricant. So the same modification can be used as a lubricant. And the second thing is that you were talking about the sharks, and yeah, there has been a lot of research to trying to understand how the shark and the coral prevents microbes to colonize, right? And I did that kind of research looking at toxins when I was in marine biology, but I don't think that nobody has trying to move that research to urinary catheters. And the main goal for urinary catheters marketing is just to put antibiotics on them. And they think that that's going to solve the problem. But if we see that if these proteins cover the catheter, that means that the antibiotics will never be in contact with the pathogen by itself, right? So the protein will cover the catheter, so the pathogen will be fine because it's not in contact with antibiotics. I don't know about that research, but it will be interesting. So far, We developed that based on a plant, because a plant repels things to attach to them. So it's called slippery surface modifications. Yeah.
Thank you. This is excellent work.
Thank you.
So can you distinguish between hospital-acquired catheter-related UTIs and non-hospital-acquired UTIs as well?
So this is a really good point, right? There are people that have a urinary catheter at home. They are the same in this case. We normally call them hospital-acquired because it's normally where you're going to receive the urinary catheter. But we have seen that nursing homes, there are many people that use these urinary catheters. And there is one study that says that patients, they arrive at similar time, they have similar disease profile, and the ones that receive the urinary catheter die faster because of all the infections that they were having than other patients, right? So we believe it's the same situation. We really believe it's because of this inflammation is allowing these proteins to attach to the catheter, and then it's what these pathogens use actually to colonize. But that will happen in a home and in a hospital. And again, like, we're not thinking about that our hands have bacteria, right? And our skin has bacteria, and those bacteria may be able to colonize at the end. Yeah.
So the fibrogen in your modified catheter is not attaching to the catheter itself, yes. But presumably the fibrogen is still present in the bladder itself.
Yes, it is.
So it seems that perhaps your modification involves two things. There was a design change to the catheter itself, and a surface change to it as well. Is that right? So because otherwise you might still be causing the inflammation that would still cause the fibrogen.
Yeah, so one of the cool things about this catheter is it's really slippery, right? So it's really soft. So it doesn't cause the same damage that a urinary catheter will cause. So indeed, fibrinogen is still, because we have done the imaging of bladders, we have seen what is the fibrinogen, where the pathogen is. The cool thing about this is that it causes less damage, and even if it's a little bit of damage, the fibrinogen will be trying to heal the damaged tissue, but it won't deposit on the catheter. And when we believe that is the main perpetrator, and we have imaged the whole urinary catheter, you see fibrinogen deposition, and it's more concentrated where it was inside of the bladder, but it keeps going down, 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 right? So that means that it's actually fibrinogen all around the catheter. So if somebody's outside and it touched that, and the bacteria is able to attach to it, it may migrate up. So we still have fibrinogen that will heal the tissue, but will prevent that these pathogens migrate up to use that fibrinogen in the tissue to colonize, because we're preventing the first step, right? Did that make sense?
It does. I'm going to have to think about it a little bit more, because it's,
yes.
It's
believe me, it caused me a lot of problems when I'm trying to understand this. Well, it was like, why nobody studies this? And then I figured it out because it was really hard. I tried to do biofilms, or like to put the catheter in urine and the bacteria to colonize, and we didn't see anything. And the important thing is that we didn't see even the bacteria was growing in urine. So it's like it was driving me crazy. Like, how can you infect if you cannot grow in urine and you cannot even attach? But it's all these proteins, right? So,
could you control for those two different changes and do the experiment where you have a very soft catheter or a modified catheter,
yes.
but fibrogen could still attach to it?
So
would that not cause the damage and then not have a problem?
Yeah. So what we are doing right now, and actually it's part of the grant that we got from NIH, is that we're modifying at different degrees to know how much we can modify, like if the modification also regulates the deposition, right? And then we can see whether if we remove 50% of the deposition of the proteins on the catheter, but the catheter is still a little bit not as soft, they will keep causing more damage. So we will see the modification percentage with inflammation and prevention of infection. So we're trying to understand what is the dynamic. And yeah, we're doing this right now, but what we know right now is that the fully infused catheter works perfectly. Yes.
It seems like those secondary experiments are going to be important for you
Yes.
to protect your patent.
Yes.
from Other people that are going to try to knock it off.
Exactly. So we're working on it, and they are really impressed. We're working with tons of urologists. They want to move this to stents now, and we can use, there are new stents, they are silicone based, and they have a peak model that we can use exactly the same stents they are using in humans because the ureter is the same size. So we hopefully can able to infuse those ones and see if we can prevent those infections too in the ureters and in the kidneys. Yeah, we're really excited. Yes. Thank you.
And let me just tell you what's coming up in the next 2 weeks. And so let me pull this up here. So in 2 weeks, we're actually going to have a panel of people. So 2022, the first week of October, in case you missed it, there were Nobel Prizes that were awarded. We're going to focus on the Nobel Prizes that were awarded in chemistry, physics, and physiology and medicine. And so we will have a panel that will be talking about each of those awards for about 10 minutes and sharing why those, what those awards were and also why they are interesting to us. And then we'll have time for discussion as well. So please join us October 25th. We'll be here. Nobel Prizes. There are 3 different topics. Should be really interesting. Everything from Neanderthals to photon entanglement. And I would also like to thank our sponsors. So the public library, so Eric in the back, and the University of Notre Dame, and as well as the College of Liberal Arts and Sciences at Indiana University South Bend. And thank you for coming tonight.